This chapter is on the cells. This will help you with your Chapter 3 worksheet. Let's begin with the discussion on what a cell is. The cell is the smallest structural and functional living unit. We talked about the structural hierarchy of organization in Chapter 1, and we learned that it goes from atoms and molecules to cells. Cells can actually live on their own, and in fact there are over 200 different types of human cells. Other types of cells include bacteria and protistans. Cell types differ in size, shape, subcellular components, and functions. The cell theory states that all cells give rise to other cells, meaning that cells do not spontaneously appear out of nowhere. They have to divide and reproduce. Here's an example of some cell types in the human body. Illustration A shows cells that connect body parts, form linings, or transport gases. These include epithelial cells and connective tissue cells. Illustration B shows cells that move organs and body parts. This shows skeletal muscle and smooth muscle cells. There's a third type of muscle cell called the cardiac muscle. Illustration C shows a cell that stores nutrients, such as a fat cell, also known as an adipocyte. Illustration D shows a cell that fights disease. This is called a macrophage, and you can see the extensions off of the macrophage. This type of cell actually moves on its own throughout the body, searching for pathogens to destroy. Figure E shows a cell that gathers information and controls body functions. This would be the nerve cell. And figure F shows a cell of reproduction. Sperm and eggs are the two cells of reproduction, and sperms can swim. Many animals release their sperm into the external environment, where they swim to the egg of their female. In humans, fertilization is internal. In the generalized cell, all cells have the following common structure and functions: the plasma membrane, the cytoplasm, and the nucleus. The plasma membrane is a flexible outer boundary that serves as the skin of the cell and also a center of communication. The cytoplasm is the intracellular fluid. That means that's the fluid inside the cell, and it contains organelles. Organelles are essentially small organs that all have a purpose. The nucleus is the control center. This serves as the brain of the cell. Here's an illustration of a typical human cell. The plasma membrane on the outside forms the skin and also serves as a site of communication. Here we see secretions being released, such as digestive enzymes, so it's also a site of very important membrane transport. The cytoplasm is the material between the plasma membrane and the nucleus, and as you can see, it's filled with organelles, small organs that all have a purpose, a function, and also a fluid component called cytosol. And then the nucleus in the center is generally the largest structure of most cells, and it serves as the control center or the cell's brain. This is where the DNA is contained. Let's start with the plasma membrane in our discussion on the three major components of the cells. We're going to look at each of those in more detail now. So the plasma membrane is a bimolecular layer, meaning it's a double layer, and it contains lipids and proteins that are in a constantly changing fluid mosaic. So the plasma membrane is not rigid. It's very fluid. It has a consistency of oil. The plasma membrane plays a dynamic role in cellular activity. As mentioned, it serves as the skin. It also serves as sites of communication and membrane transport. As the skin functions, it separates the intracellular fluid, or the fluid inside the cell, from the extracellular fluid, or the fluid outside the cell. Another name for extracellular fluid is interstitial fluid, also known as tissue fluid. The bimolecular layers, are made of lipids and proteins. Here are the lipids. 75% of the lipids are phospholipids. And so you'll commonly hear the plasma membrane called the lipid bilayer. 5% is glycolipids, which are lipids that have a sugar attached to them, and 20% is cholesterol. So the phospholipids are the main structural lipids in the cell membrane. The glycolipids help to maintain structure and also anchor some membrane proteins, and the cholesterol increases membrane stability and fluidity. Because cholesterol is waxy in its structure, whereas, as mentioned, the plasma membrane is like an oil in its consistency. And so the cholesterol helps to hold everything together and give it a little more form. There are also proteins in the plasma membrane, and the proteins include integral proteins, which are going to run through the entire surface to middle to inner surface of the plasma membrane. Peripheral proteins, which will be loosely attached to the integral proteins and found either on the outside or the inside of the membrane. And then a structure called the glycocalyx. The glycocalyx is composed of glycolipids and glycoproteins. Essentially, it's a bunch of sugars that are connected to either fats, which are lipids, or proteins. This illustration shows the lipids and the proteins of the plasma membrane. The phospholipids make up that lipid bilayer. The circular component is the phospho part, and the wiggly tail component is the lipid part. Notice that the lipid part is in the center of the membrane away from water. Oil and water don't mix, and so the lipids are protected from the watery external environment and internal environment of the cell by the phosphate heads. That's why it's a double membrane. This is the cholesterol in yellow, and it's waxy, and you can see that it is anchoring the lipids and some proteins in place. Integral proteins go all the way through the membrane, from the outside, middle, and inside. Peripheral proteins are anchored to integral proteins and are either on the outside or the inside, and they generally act as enzymes, whereas integral proteins can act as channels. As you can see here, substances can go through the membrane through this integral protein. On the outside is the glycocalyx, and wherever the carbohydrates are attached to a protein, that's a glycoprotein. Wherever the carbohydrates are attached to a lipid, that's a glycolipid. In a sense, your cell membrane is covered in a layer of sugar. Sugar and spice make everything nice. And so the sugar is very important in that communication property that I mentioned earlier. because the glycolipid and glycoproteins of individual cells are unique to the person that they belong to. Here are the different kinds of membrane proteins. Similarly, like I mentioned before, we have transport proteins. And so the protein to the left here is going to form a channel that will let a particular solute or substance pass through. Some, like the one on the right, actually require energy and serve as pumps because they will pump the solute or the substance against a concentration gradient. And these are integral proteins, to answer the starred question. Another function is as a receptor for a process called signal transduction. This is very important factor in the way hormones work. When a membrane protein exposed to the outside of the cell has a binding site with a specific shape that fits the shape of a hormone, the external signal may cause a change in the shape of the protein, and this initiates a change or chain of chemical reactions within the cell. And that's how hormones get things going in the cell. And this also is an integral protein. Another function is attachment of a skeleton inside or outside the cell. So on the outside of the cell, the extracellular matrix will be proteins like collagen. Collagen is actually the most abundant protein in our bodies, and it binds all the organs and tissues of our bodies together. And so this would be collagen in the extracellular matrix, most likely. And this protein is attached to it, anchoring that cell within your body. In the intracellular matrix, the internal environment, we call this the cytoskeleton. And this will be comprised of a protein of actin or something similar, but not collagen. In it, anytime you see collagen, it is extracellular. And enzymes. I mentioned earlier that some peripheral proteins will act as enzymes. Here it looks like an integral protein is acting as an enzyme. And so an enzyme is going to be able to catalyze a step of a chemical reaction. It basically is going to take product or substances, reactants A and B, and make a product C, or it can take product or reactant C and then break it apart into A and B. That's how an enzyme will work. It will have an active site that these molecules can attach to, and the chemical reaction takes place there. So it's a reactor, essentially. Cells are not only joined to collagen and a cytoskeleton, but they're also joined to one another. And in this case, we see a membrane protein called a CAM joined to another membrane protein in an adjacent cell. And this will connect the cells together and is very important in certain tissues like epithelial tissues that form linings and boundaries of organs. As mentioned earlier, the glycocalyx is important in membrane communication and how your glycocalyx in your body is unique to you. And your cells in your immune system will be able to recognize the glycocalyx of your body as self. And that helps the immune system not attack your own body cells. Whereas a foreign cell would come in with a glycocalyx that's foreign to the immune system and thus will be attacked. If the cells of the immune system malfunction and they think that your own cells are pathogens, that is called an autoimmune response. Since we are multicellular animals, our cells are going to be closely associated with one another. And as such, most cells, are actually going to be joined together. Not every cell is an immune cell like that macrophage going around and looking for pathogens. Most cells have to make up the next level of structural hierarchy, which is the tissue level, which then builds our organs, etc. So to join the cells together, we have to have junctions. Tight junctions are going to be found in epithelial cells, as mentioned earlier, that form linings and boundaries of organs. Desmosomes are going to be components of the cytoskeleton that give flexible support to adjacent cells. And gap junctions are going to be porous, and so substances can flow freely between cells. This is very important in cardiac muscle cells that rely on ionic exchange as the cardiac rhythm commences. In the illustration at top, it shows epithelial cells that are forming a boundary or a lining of an organ, and they have to be tightly connected to one another through these tight junctions. Notice that they kind of look like they are nailed or stapled together. The desmosomes are the next one here, and they are structures in the cytoskeleton that give flexible support. So the cell, is obviously going to be moving around as the organ is moving, because organs are not stationary. They're always doing some kind of movement. And so this will give flexible support, kind of like guy wires or steel cables on a bridge. And then the third type shown in this picture is the gap junctions. And you can see that there's pores or channels between them where ions can flow from cell to cell. We'll generally see gap junctions in the cardiac muscle cells. Different types of membrane transport. Solutes and water are selectively transported across the cell membrane. Passive transport does not require ATP, and substances move down a concentration gradient. Active transport requires ATP, and substances move against a concentration gradient. This illustration shows concentration gradients and passive and active transport. On the left shows passive transport, where substances are moving down their concentration gradients from a higher concentration to a lower concentration. In active transport, the substances are moving from a lower concentration to a higher concentration, so they use ATP in order to power that pumping action. In passive transport, substances move down their concentration gradient. This does not require ATP because the concentration gradient carries the solutes along as if moving down a slide. There are two major types of passive transport: diffusion and osmosis. Diffusion is the passive transport of solutes. Osmosis is the passive transport of the solvent, which is water. Diffusion can be classified as simple or facilitated. Simple diffusion transports nonpolar molecules directly through the plasma membrane. Examples of molecules that can go through the plasma membrane are oxygen, carbon dioxide, and certain metabolic wastes. Water is an exception. It can undergo simple diffusion even though it's polar, meaning it has charges. Facilitated diffusion transports ions and polar molecules through a transport protein. The transport protein has a hydrophobic exterior that interacts with the phospholipid tails. Remember, oil and water don't mix, and hydrophobic means that it doesn't like water, but it will be able then to interact with the lipids or the fats. The transport protein will have a hydrophilic or water-loving interior that interacts with the ions and the polar molecules as they pass through the plasma membrane. Examples of molecules that undergo facilitated diffusion are glucose, sodium, and chloride. Simple diffusion of molecules directly through the phospholipid bilayer is pictured in this illustration. These small nonpolar molecules are diffusing directly through the cell membrane. Facilitated diffusion has to use a channel protein in order for polar particles to pass through. Sometimes the molecules are very large and the protein carrier has to change shape. No energy is required because it's still moving down a concentration gradient. Osmosis is the diffusion of a solvent, such as water, down its concentration gradient. I've learned that water follows salt. So if you think about that, you can get an idea of what is happening in the next few solutions we're going to talk about. Cells live in several different types of solution based on how salty they are. And the water will follow the salt, and this causes tonicity to be affected. Tonicity is the ability of a solution's solute concentration to cause a cell to maintain shape, shrink, or swell. The three solutions found within the body are isotonic, hypertonic, and hypotonic. In an isotonic solution, it is a solution that has the same amount of salt as the inside of the cell. An isotonic solution is equal to 0.9% saline or 5% glucose. The cell is able to maintain function in a body because it can maintain its regular shape. Remember, structure influences function. In a hypertonic solution, the solution has greater solute concentration than the inside of the cell. Hyper means above. So this is a salty environment. Water follows salt, and so water will be pulled out of the cell and into the solution. A hypertonic solution is measured as greater than 0.9% saline or greater than 5% glucose. As a result, the cell shrinks or crenates and the function is impaired. A hypotonic solution has less salt than the inside of the cell. Water is pulled out of solution and into the cell as a result. A hypotonic solution is less than 0.9% saline or less than 5% glucose. This cell will swell up as it fills with water and may burst or lyse. In active transport, substances move against their concentration gradient. This requires ATP because moving against the solute concentration is like moving up a slide. It's going against gravity, basically. There are two major types, primary active transport and vesicular transport. In primary active transport, it uses ATP as the source of energy to power protein pumps. Vesicular transport uses ATP to bring solutions into the cell via endocytosis, or to export them out of the cell via exocytosis. The sodium-potassium pump is a common example of primary active transport. The sodium-potassium pump will export 3 sodiums out of the cell and import 2 potassiums back into the cell. More sodium is concentrated outside of the cell, and more potassium is concentrated inside the cell. This forms the resting membrane potential. The inside of the cell membrane has a negative charge relative to the outside of the cell membrane. This powers nerve impulses and muscle contractions. It makes the cell membrane a battery. In step 1, the sodium-potassium pump binds 3 sodium ions and a molecule of ATP as it's open to the inside of the cell. In step 2, the splitting of ATP provides the energy to change the shape of the channel. The sodium ions are driven through the channel as the pump opens to the outside of the cell. In step 3, the sodium ions are released to the outside of the membrane, and the new shape of the channel allows 2 potassium ions to bind. In step 4, the release of the phosphate ion allows the channel to revert to its original form, releasing the potassium ions into the inside of the cell membrane, or the cell. Notice that the pump is now open to the inside once more, and the cycle can repeat itself as 3 new sodiums will come in and bind to the sodium-potassium pump, as will a molecule of ATP. Vesicular transport uses the plasma membrane to move bulk amounts of solutes into, out of, and throughout the cell. Exocytosis occurs when a cell secretes products or expels wastes. Endocytosis occurs when substances are transported into a cell. Exo means outside, cyto means the cell, endo means inside, and cyto means the cell. Exocytosis occurs when a cell secretes a product such as hormones, antibodies, neurotransmitters, mucus, sweat, and oils. It can also be used to excrete wastes. Endocytosis occurs when substances are transported into a cell. There are 3 types of endocytosis. Phagocytosis occurs when a cell engulfs a large particle such as bacteria. This is the function of macrophages, which are a type of white blood cell in the immune system. Penocytosis occurs when a cell takes in solutes from a solution. This occurs in the intestinal cells when they absorb nutrients from your food. Receptor-mediated endocytosis occurs when a cell brings solutes in at the direction of a hormone. So this would require the membrane receptors, as seen earlier, and then a chain of chemical events will occur, such as when insulin binds to a cell, glucose will be absorbed. Picture on the left is of phagocytosis, when a white blood cell is engulfing a bacteria to be destroyed. The middle illustration is penocytosis, that occurs in the intestines, and a solution of sugars and other small particles from your food are absorbed. And receptor-mediated cytosis is pictured on the right as the hormone binds to the receptor on the cell. It allows for the substances to enter, such as insulin binding to the receptors and then glucose entering. The second major part of the cell is the cytoplasm. Remember, the human cells have 3 major parts: the plasma membrane, the cytoplasm, and the nucleus. We just concluded our discussion on the plasma membrane. Now let's look at the cytoplasm. The The cytoplasm is located between the plasma membrane and the nucleus. It houses two major types of organelles, membranous organelles and non-membranous organelles. Membranous organelles have a phospholipid bilayer just like the plasma membrane, and they include the endoplasmic reticulum, the Golgi apparatus, peroxisomes, lysosomes, and mitochondria. The non-membranous organelles do not have a lipid bilayer around them, and they include ribosomes, cytoskeleton, and centrosomes. The endoplasmic reticulum is continuous with the nucleus. It has two regions, a rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough endoplasmic reticulum is studded with ribosomes. Ribosomes synthesize proteins, which are transported into the rough endoplasmic reticulum. The smooth endoplasmic reticulum lacks ribosomes, and it is the site for lipid synthesis used in cell membranes, fatty acid breakdown, and toxin degradation. Ribosomes are where proteins are made, and they consist of two subunits of ribosomal RNA and proteins. The reason that the rough endoplasmic reticulum is attached to the nucleus and contains a bunch of ribosomes is because the DNA encodes for the proteins that will be made by the ribosomes. This illustration shows the nucleus and the endoplasmic reticulum. The little red dots are ribosomes. The rough endoplasmic reticulum is continuous with the nucleus, and it's studded with ribosomes that will synthesize the proteins as they are transported from the DNA into the rough endoplasmic reticulum. The smooth ER lacks ribosomes, and it will not synthesize proteins. Instead, it will synthesize lipids that will be part of the membranes. It will also detoxify substances, and it's important in cholesterol synthesis too. The Golgi apparatus is going to work on the proteins modified in the endoplasmic reticulum. The Golgi apparatus will put sugar side chains on the proteins and any lipids, and also cleave some proteins, snipping them and cleaning them up for transport. Basically, they're going to finish the proteins into their final products, pack them up, and ship them out in vesicles to be used as secretions like hormones or antibodies or sweat, oil, things like that. Vesicles are sacs that form as buds from the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane. They function as storage centers or transport agents. A lysosome is vesicle that bud from Golgi bodies and take part in intracellular digestion. They help to break down dead and dying organelles, as well as participate in phagocytosis as a bacteria is being broken down. Peroxisomes bud off of the endoplasmic reticulum, and they hold enzymes that digest fatty acids, they digest amino acids, and they also have hydrogen peroxide that will help to degrade alcohol and free radicals, which are going to be toxic substances that can damage the cell. Altogether, this forms a organelle system called the endomembrane system. I mentioned earlier that organelles are little organs inside a cell that have a particular function, a purpose. And not only are the organelles like little organs, but they also work in a system. So remember, the cell is the smallest living unit of life. It's a little microcosm of the way the entire human body works. So starting with number 1, you can see the rough ER would be attached to the nucleus, and it says protein-containing vesicles will pinch off of the rough ER and migrate to fuse with membranes on the Golgi apparatus. In step 2, proteins are modified within the Golgi compartments. They're got sugars added to them, or they're folded up, or they're cleaved. And in step 3, the proteins are then packaged within different vesicle types depending on their ultimate destination. In pathway A, the proteins are going to undergo exocytosis. As mentioned, there'll be a hormone or an antibody or sweat or oil or digestive enzyme. In pathway B, the vesicle membrane will be incorporated into the plasma membrane to actually help rebuild and repair the cell membrane. In pathway C, it will become a lysosome, which will contain acid hydrolase enzymes, which will basically be stomach acid-like substances that will digest old and dying organelles as well as any pathogens that like a macrophage would eat up. The mitochondria. Everybody knows the mitochondria. This is the energy powerhouse of the cell. It specializes in ATP formation and is the major organelle in cellular respiration. The mitochondria is kind of joked about because it is so well known, but you are going to really gain an appreciation for just how important this organelle is, because the ATP that is needed to move our proteins for active transport, to move our proteins for muscle contraction, is astonishing. It takes about 10 million molecules of ATP per muscle per second to move a bone in your body to help you manipulate the environment. 10 million molecules of ATP per muscle per second. So these little guys have their work cut out for them to power us and keep us going. And it's interesting because in size and chemistry, mitochondria resemble bacteria. They have their own DNA, separate from ours. They divide on their own. They don't rely on our cells' mitosis for division. And they have their own ribosomes, so they can do their own protein synthesis. It is thought that the mitochondria used to be a free-living bacteria out in the environment on its own and was engulfed by ancient cells and formed a symbiotic relationship, thus giving the mitochondria a home and nutrients and a safe place to divide and do all of its work, and then giving the larger eukaryotic cell the ATP necessary to do what it needs to do in the environment, such as move and grow and reproduce. Win-win. Another non-membranous organelle, we looked at ribosomes already, which were sites of protein synthesis. Another non-membranous organelle is the cytoskeleton. So again, the cell is a microcosm that shows just the smallest unit of structural organization of an actual living thing. It has organelles, it's got skin, and it even has a brain, which we'll talk about next, in the nucleus, and it even has a skeleton. So the cytoskeleton is going to contain 3 major components: the microtubules, microfilaments, and intermediate filaments. Of the 3, the microtubules will be the largest cytoskeletal elements. They are made of tubulin subunits, which is a protein, and this helps keep the organelles and cell structures in place or move them to different locations. Looking at the endomembrane system, these vesicles aren't just floating from the rough ER to the Golgi apparatus. They are actually being transported, they're being walked along the microtubules by a transport protein. That is also using ATP. You can see the microtubules in the illustration here. They're these ghostly-like white structures. And here is the transport protein that is walking the vesicle along the microtubule. Microfilaments are the thinnest of the cytoskeletal elements, and they are made of actin chains, which are composed of little actin subunits. You're going to learn a lot about actin when we get to the muscles, because actin helps the muscles contract. And that's mentioned here under the functions of microfilaments, as is reinforcing cell shape, helping to cleave the cell in division, and anchor membrane proteins. The third type of cytoskeleton is the intermediate filament. This helps strengthen and maintain the shape of cells or cell parts. It's kind of the network that occurs under the plasma membrane. There's an interesting organelle that occurs only in animal cells, so it won't be found in plants or bacteria or fungi, and it's called the centrosome. And this helps to generate the microtubules and organizes the mitotic spindle fibers that are formed during cell division. This is the illustration of the centrosome and the microtubules that are forming. Another thing that microtubules will do is they will form cellular extensions. So remember, microtubules are the largest of the cytoskeletal elements, and we saw the picture of a transport protein walking a vesicle along it. And so these large cytoskeletal elements are actually hollow, and they are able to move and whip around, so they can form the cilia and the flagella. This also requires ATP in order for these cellular extensions to move. Another cellular extension that's not part of the cytoskeleton, it's actually part of the plasma membrane, are the microvilli. Microvilli are finger-like extensions of the plasma membrane that increase the surface area for the absorption of nutrients during penocytosis. It has a core of actin filaments to help support them. Now let's talk about the nucleus, the third major structure found in the human cell. So we discussed the plasma membrane, we just went through the cytoplasm, and now let's take a look at the nucleus. I'm turning a page of my study guide. Okay, so the two major functions of the nucleus is to keep DNA from getting tangled in cytoplasmic machinery and isolate it from damaging reactions, like when hormones, are causing a cascade of chemical reactions, the DNA will be protected from that. The outer membrane of the nucleus is a boundary that controls the movement of the substances to and from the cytoplasm. And remember, the nucleus is attached to the endoplasmic reticulum. One of the big things that's going to be leaving the nucleus is RNA. And RNA is made in the nucleolus, as seen as one of the four main components of the nucleus. And the nucleoplasm is going to be the liquid inside, and of course the nuclear envelope is the plasma membrane that is surrounding the nucleus. And the DNA is going to code for the proteins. The way that it works is it codes for what's called a messenger RNA, and that RNA is what's able to leave the nucleus and bind to a ribosome where protein synthesis will occur. Here is a picture of the nucleus attached to the rough endoplasmic reticulum. Remember that the ribosomes are pictured as the little red dots, and that's where the site of protein synthesis is actually going to occur. The nuclear pores are little gap junctions that will allow for the movement of messenger RNA from the nucleus into the cytoplasm, where it can then bind to the ribosomes for protein synthesis. The nucleolus is presented in the center of the nucleus here, and that is where RNA is made. So as the cell goes through its lifetime, it goes through a cell cycle, and the green shows the interphase, and the yellow shows the mitotic phase or mitosis. During interphase, the cell is undergoing growth and DNA synthesis occurs once the cell has committed to going into the mitotic phase, which is cellular division. What keeps the cell cycle in check? How does the cell maintain, proper life stages? So subphases of interphase in different species of eukaryotes have specified time intervals. And one subphase must end before another begins. And this is governed by control mechanisms that occur in the G1 subphase. You can see the G1 subphase is the growth phase. Some cells will stay in the growth phase forever, like nerve cells. Once the nerve cells are formed in your cute little baby body, they will elongate as you grow, but they will never go through mitosis. If a cell, though, goes into what's called the S phase, it has committed to going through mitosis. The S phase is going to be the DNA synthesis phase, and then it will go through G2, which is going to be growth and final preparations for divisions. Some cells are dividing all the time, like epithelial cells of your skin and your organ linings. They're constantly being abraded and killed by, the environment that they are exposed to. And as such, they have to replace themselves a lot. And so those cells are going to bypass the G1 growth stage into the S phase, G2, and mitosis very regularly. Red blood cells are another example of that. In the bone marrow, they're constantly going to be undergoing mitosis and replenishing, as a blood cell only lives for about 120 days before it dies. So if you didn't continuously make more, then you would run out of blood and you too would die. So once the cell goes through G2 and has passed all the checkpoints, it goes into mitosis. Mitosis is nuclear division, and it has 4 stages: prophase, metaphase, anaphase, and telophase. In prophase, the duplicated chromosomes from DNA synthesis are going to condense and look like the X-shaped structures that you're familiar with. The centrosome is going to move its little centrioles to the opposite poles of the cell, and the microtubules will be assembled so that the spindle fibers can form. In order for the microtubules to attach to the chromosomes and move them apart from one another, the nuclear envelope has to break up and dissolve. In metaphase, the chromosomes are attached to the spindle fibers, and the spindle fibers are going to line them up in the equatorial region of the cell, so along the midline of the cell. Notice that the spindle fibers are attached to specific parts on the cell. These are called kinetochores, and they are going to help move the chromosomes apart from one another. Remember me telling you there's transport proteins that walk vesicles through the cells using the microtubules as their highway? The same thing is happening here. And once the chromosomes start to move apart from each other, we enter anaphase. So the sister chromatids, which are the, half of the chromosome, are pulled apart from each other. And once separated, each chromatid is now an unduplicated chromosome, and the cell can start to cleave apart and divide into 2 daughter cells. And that occurs in telophase. So once the daughter cells start to form, the chromosomes can decondense or unravel and go back to their relaxed DNA state, and 2 new nuclear membranes will form around the new sets of chromosomes that were made, and you see that there starts to become a cleavage furrow where the cell is pinching inward, and it will eventually be pinched apart into the 2 daughter cells. Cytokinesis is the actual pinching apart. The cell is going to start to use those actin filaments to kind of tie the plasma membrane together, because we don't want the cell to ever be open to the outside world, because that would kill the cell. It'd be like it, got ripped open. And so we need to use the actin filaments to, kind of squeeze the ends of the membrane now together and pinch them off, just like you would tie a string around a piece of fabric. So in step 1, mitosis is over and the spindle is now disassembling. In step 2, at the former spindle equator, a ring of microfilaments, the actin, is attached to the plasma membrane and it contracts. In step 3, as its diameter shrinks, it pulls the cell surface inward. And in step 4, as the contractions continue, the cell will be pinched into two. So once the daughter cells form, they're actually smaller than the parent was, but they will grow. And each of the daughter nuclei will have the same chromosome number as the parent cell did. And so as it grows, remember the smooth endoplasmic reticulum will be making lipids that will help to replenish the membrane, and also vesicles that are going to be made in the Golgi apparatus will also help to replenish the membrane. So the cytoplasm grows, organelles will grow, and the plasma membrane can grow. Because, again, the cell is a microcosm of the body system. It's the smallest structural unit of life. And so it can grow and reproduce just like a human does. And we actually grow and reproduce through mitosis of our cells, the growth and reparation, and then meiosis, which is how eggs and sperm are made. And that is the end of chapter 3. Thank you for listening.